Yeah.
Yeah. It's just like more satisfying than that point. More satisfying would be, there's a PDF great for finding something quick.
Timer.
For when we read.
If he leaves me, should I go?
To read a print.
I do.
I would have been okay with your face on the screen the whole time, just to be clear. But, yeah. Yeah. Are we good? Okay. Dave, you want? Oh.
Hello, check.
There we go.
There we go. All right. Well, welcome. Talk number one of a series of many talks going on throughout the month. And this is, you all know Sam. He's an institution here. He was an intern here. He was a visiting researcher here that was then kind of trapped somehow for a while here because of COVID, I think. Now he's back as a postdoc. And yeah, I mean, between that, he did a PhD at Trinity College, Dublin. And then he actually came here possibly illegally because he hadn't gotten his PhD yet. And there was, some drama surrounding that. So that's fun. Anyway, so as the title suggests, this is part one of 8, 9, 10 part series throughout the month. Sponsored by the Theoretical Visiting Sciences program. This is kind of their first demo for this long or sort of short-term visiting workshop thing that seems awesome. So if you're not within this workshop and maybe an onlooker, definitely something to consider putting together yourself. So Sam's going to introduce his research today, and then Owen will be talking right directly afterwards. So don't leave after this talk, please. 2 great talks today. So yes, please take it away.
Thanks, Dave. I'm going to get Dave to MC all my talks. That was great. So thanks, everyone. As Dave said, I'm kicking off that's me. A series of talks on stability and response diversity that is funded through the OIST TSVP thematic program, which I'm really excited about. I'm from Dave's lab. I'm in the Integrative Community Ecology Unit. And I also put integrative in the title of my talk, just to hammer that point home, that we really try and get at key ecological questions using a kind of variety of complementary approaches. So I want to start, before anything else, by thanking Jonas and the whole TSVP team for making this whole thing happen. I'm really excited that we're doing this. And we'll be talking about buckets and microphones throughout the day today. So another title to this talk could have been, what can buckets and microphones tell us about disturbance and stability in the natural world? So I'm going to start with some background and then we'll move on to the bulk of things. So what is stability? Well, it's really the central framework for thinking about how ecosystems respond to environmental change. And the concept is complex and multidimensional. There's lots of different ways that you can measure stability or think about stability. So if you have some ecological variable of interest, maybe you're interested in the population of a species or an ecosystem function or service, then you can look at how it varies through time or over space. Or if you have some known perturbation, some disturbance, then you can also measure the change in that variable after that disturbance as, say, resistance to or recovery from that disturbance. Think about the insurance effect of biodiversity, which is a pretty long-standing kind of framework now. We know that biodiversity both increases and stabilizes aggregate kind of emergent properties of ecosystems like ecosystem functioning. We see in this classical example, as you increase the number of species in these simulations, not only does the mean value of productivity, simulated productivity, go up, but the fluctuations around that mean dampen. And that's actually the opposite of what we would expect based on purely statistical scaling. So for several decades now, ecologists have been interested in what the biological mechanisms might be that explain that kind of counterintuitive notion. And so we've all seen pictures like this, we know that humans are modifying the planet at an unprecedented rate and scale. And I really quite like this quote that says, disturbances and stability are multidimensional, but our understanding of them is not. And so that's really the inspiration behind a lot of what I and many people do in this space. And so, as I mentioned today, I'm going to talk first about some buckets, then about some microphones, and then we're going to move on to the concept of response diversity, which is the topic for our thematic session over the next few weeks. So I'm going to start with this field experiment in Tomakomai Experimental Forest, which is in Hokkaido. And it was a collaboration with Hokkaido University. And we know that under the climate crisis, climate change is increasing mean temperatures, but it's also resulting in many more of these extreme events, things like heat waves. We're seeing more heat waves, they're getting more intense, they're lasting longer. And so it's really important that we understand how ecosystems respond to these extreme events. And at the same time as the climate crisis, we're also facing a biodiversity crisis. Species are being driven to extinction at rates that are thousands of times beyond expected background rates of extinction. And typically those extinctions tend to be non-random. If you imagine kind of a classic food chain, it tends to be the predators that go extinct first, and then their prey and so on down the food chain. And so today I'm going to be talking about an experiment where we were specifically looking at the effect of a predator. And this is that guy. This is a freshwater sculpin, which is a fish that was swimming around some buckets, basically, and eating things. And so this is a mesocosm experiment because we use buckets. In this case, this was they were kind of this big. It was a cake tin, where we can use these mesocosms as kind of a middle ground between, say, a fully controlled laboratory experiment and some completely natural observation in the field. A mesocosm tries to bridge that gap as being a kind of semi-controlled but semi-open field experiment. And so in this example, we were looking at the combined effects of heat waves and species extinctions. And so we had half of our mesocosms had this predatory fish, and half of them didn't. And so that was meant to be the kind of extinct scenario. And so this is what that looked like. So this is the Horonai stream that runs up the left. We've got a pump here that pumps water in from upstream. It goes into these 3 header tanks here. And then gravity feeds the water through down into these small, as I say, small buckets, where it then flows through and out the center and back into the stream. So it is a kind of pretty open system. Not everything like the fish, obviously, is too big to escape. But it allows kind of colonization and drift of macroinvertebrates and algae and diatoms and things, bacteria. So that's what this looks like. We've got these header tanks and we've got these 48 mesocosms here. And so we wanted to compare the effect of heat waves in the presence and absence of the predator to see if the predator can modify heat wave impacts. And so to make these heat waves, it's a fairly simple setup, actually. It was just a gas boiler system. So some of the water was being fed through the boiler and it was being heated. And then there was also unheated water. We were mixing it together to get the temperatures that we wanted. And that was pretty tough work, actually. We had to manually adjust the temperatures every hour, even through the night. So that was a pretty rough 10-day period. But we ended up with these 3 different scenarios. We had the ambient temperatures that were just what we saw during the experiment. There was no manipulation of temperature in one of the 3 scenarios. We also had the current heat wave scenario, which was about 2.5 degree heat wave, which is based on observed heat waves for the past century or so, based on historical data. And then we also had higher intensity, 6-ish degree heat wave based on the IPCC's worst case scenario for climate change projections for the local Tomakomai region for the end of the century. And so the point with this was that we were trying to get these be locally relevant. So they were specifically what we expect what this stream to do or be exposed to currently and in the future. So what did we find? Well, unsurprisingly, the predators had an important effect in the system. So we saw that where they were present, so this little blue fish here is predator present, they were reducing macroinvertebrate densities simply because they were eating bugs and macroinvertebrates in the system, right? And so they were lowering densities that way. And then that had nice knock-on consequences for leaf litter processing. That's an ecosystem function by these macroinvertebrates. So because there were fewer macroinvertebrates there to eat the leaves, they had this trophic cascading effect. And they also shifted the macroinvertebrate communities because they were selectively eating certain species over others. But we weren't just interested in the fish on their own. We wanted to see what they did, how that might affect the response of the system to heat waves. And so we took daily measures of algal biomass and composition using this bentho torch, and we could do that in multiple microhabitats within each mesocosm. So we could get a measure of spatial variability as well as a measure of temporal variability through time. And so, of course, there were lots of results and they were a bit nuanced. So I'm just going to focus on one key point, which is that the heat waves shifted the composition of algae in the system. And they also homogenized the algal community in space. So that is, after the heat wave, the different microhabitats got more similar to each other in terms of composition. And that's actually a loss of stability, because when we're thinking about compositional spatial variability, if you have different species in different places, you're more likely to have these kind of insurance effects, spatial insurance effects, rescue effects, and patch dynamics. So we saw this decline in spatial variability relative to the control treatment, but only where the predator was absent. So where the fish was present in the system, there was actually no significant change at all based on the heat waves. There's no heat wave effects that we found. And more recently, these are maybe the things that I'm more famous for at Oyster. We did this bucket experiment up near the gardens. That's the gardens in the background there. So there's a nice big gravel lot. And we were interested in, so these were 100-liter buckets, much bigger than the previous ones. This is a pond system, so it's still water, not stream water, like flowing water. And we had these different communities of floating macrophytes, these aquatic plants. And we were exposing these buckets to nitrogen stress. And the goal was to ask whether different growth responses in these different species in combination can stabilize the total biomass output from the buckets. And so it's a very basic question actually in response diversity that I'll come back to later on. And so we don't have any results yet. We're at the data processing stage, and we're working with Alex Mihai, who's right there, to try and automatically get abundance or biomass from these images using a template matching approach. So hopefully in this first example, you've seen that we can use buckets to ask questions about disturbance and stability. And in the first example, we saw that predator extinctions can potentially worsen the effects of heat waves on ecosystems. But that was still a pretty controlled system. Those were experiments. But how can we measure stability and disturbance in fully natural settings, in complex natural ecosystems? Well, we need high-resolution biodiversity data to be able to do that. And one promising tool for getting at that is acoustic monitoring. And so when we think about acoustic monitoring, we need to think about the soundscape, which is the set of all observable sounds in an ecosystem. And the soundscape is composed of three main components. The first one being biophony, which is all vocalizing animals like these birds. I'm actually going to do this because I'm not sure if my thing's plugged in. Yeah. There's also geophony, which is all natural but non-biological sounds like wind and rain. And there's anthropophony, which is all human-related sounds, like this aeroplane. And one major advantage of these acoustic data that we're increasingly getting access to now is that they can really give us really quite high resolution data, both through time and across space, which allows us to ask actually pretty basic fundamental questions in ecology, even though this acoustic monitoring, as the name suggests, was developed for kind of conservation monitoring and applied ecology. We can also ask, I think, pretty cool questions about, for example, disturbance and stability and recovery. And so I'm now going to demonstrate some of those ideas using Okinawa as a case study. So normally I introduce where Okinawa is, but since we're all here, I probably don't need to do that. People regularly ask me why I study Okinawa. Well, I mean, it's pretty obvious, really. When you come from rainy England, it's a nice place to be. But more importantly, it has this really strong rural to developed land gradient. So there's a lot of development, particularly in the south. So there's about 1.4 million people in Okinawa, and they almost all live in these blue urban areas, particularly mainly in the south. Of course, there's Nago up there, but most of the land development is in the south. And so this means that we can ask really important questions about how land use change and human activity affects ecological communities. And so this is what our recording setup looks like. These are Wildlife Acoustics SM4 monitors, and we go out and we strap them on trees, and we've been doing that since February of 2017. So yes, I have been coming here since I had hair. So this is, we've got one of these recorders on each of these trees across 24 different sites. So this acoustic monitoring work is part of the wider Okion Churamori project, which is the Okinawa Environmental Observation Network that was set up by urban economer Masashi Yoshimura in the early days of VOICE. So at each of these sites, we've been recording for originally 10 minutes out of every half hour. Now it's 1 minute out of every half hour. But we've been doing that since February 2017, which means that right now we have more than 400,000 hours of continuous audio data. So to put that into perspective, if I wanted to listen to it all, it would take me more than 50 continuous years without a break. So it's a lot of data that we have to find a way to deal with. And so we can take this acoustic data, they're basically MP3 files, they're WAV files, and we can pull them in and turn them into spectrograms like this. And you would read this how you would read a musical score. Time moves from left to right, and you've got higher frequency, higher pitched sounds nearer the top of the y-axis. And we can get ecologically useful information from the soundscape because species signal their presence at a given time and location, and they have these characteristic call types. You can identify different and individual species from the soundscape. So here are some different birds singing at different times and with these characteristic signals. So Owen, this is the one you were talking about the other day. You can see the kind of so yeah. But of course, we've got too much data to sort through this all manually to find these species. So we need a kind of automated approach. So we've been using supervised machine learning in Kaleidoscope Pro, which is just a software package that comes with the monitors, basically. And so we can extract information on species dynamics from the data that we get by using this approach. So this is a 2-week period in 2016, a test case for this one species, the ruddy kingfisher, at one site. And you read this clock as a kind of almost a spiral out from the middle. So days advance out from the center. It's a 24-hour clock. So we've got midnight at the top, midday at the bottom. And the size of the bubble is just the number of vocalizations of this species detected at that site. So we can see that this kingfisher is calling mainly between about 5 o'clock and 7 o'clock in the morning. And we did this initially for 5 kind of test species across 5 different field sites. And the species represented a range of kind of habitat sensitivities or rarities. So we've got the common bulbul and crow there, and we've got this migratory kingfisher, a forest specialist owl, the Ryukyu scops owl, and then the Okinawa rail, which of course is the kind of main bird that everyone likes to talk about in Okinawa, which is endangered, and is typically found only in the Yambaru forest to the north. And we saw generally that different species varied in their detectability across the different sites, but that our more sensitive species were not detected in the more developed sites. And so this was just some very preliminary evidence that perhaps land use might be filtering the communities here and affecting which species might be present. But it's not just individual species that we're interested in. We can also use what are called acoustic indices to get a broader picture of the soundscape as a whole. So just like you might condense biodiversity down into a single number like richness or evenness, we can do the same by taking the acoustic distribution the distribution of acoustic energy of these files, and then you can do all kinds of things with them. So we might be interested in, for example, the ratio of biological to anthropogenic sound. And anthropogenic sound typically occurs at these lowest frequencies. So you can see the signal of human noise pollution on the right here. And so we can do this at very high resolution, and then we can see patterns emerge from that data. So this is 2.5 years worth of those 10-minute values, those 10-minute summaries, using that ratio of anthropogenic to biological sound. So here the brighter colors are more biological sounds. And we can see these patterns coming out. So we can see, for example, the dawn chorus in the morning when all the birds start to sing and how it shifts throughout the seasons. We've also got these really bright patches of green in the summer, which for anyone who's spent any time in Okinawa in the summer will know, of course, are cicadas. And so that was kind of an example showing that we can make inferences about different species and different aspects of biology from these broad acoustic indices. But now I'm going to show you an example where we combine individual species detections with those acoustic indices to try and understand a recovery from a disturbance. And the disturbance is this pretty nasty typhoon that hit us in 2018. If anyone was here at the time, probably remembers that was one of the big ones. So yeah, this was a 58, completely flooded up to this car. It was like a real rough one. But yeah, so we were hit by that was Typhoon Trami, and then immediately 1 week later by a Category 4 typhoon. That was Category 5 super typhoon, then immediately by Category 4 Kongray, and both went almost immediately past Okinawa, and so walloped the island completely uniformly. And so these were really big disturbance events for Okinawan ecosystems. And we can use acoustics to detect typhoons in the soundscape. So this is now just an average summary of the average volume over time. And unsurprisingly, the heavy wind and rain is really obvious in these recordings. It was so loud compared to everything else around it that it just completely distorts the graph. And a very preliminary look at the data almost immediately after the typhoon happened suggested that, so this is a typhoon here, suggested that the number of detections of this crow species seemed to be higher before than after the typhoon on average. And that got us wondering about whether there might be kind of lasting effects of these typhoons on individual species. And so one of the things I did when I joined Dave's lab was to expand this to 3 different species and across all of our field sites and give this kind of a more thorough analysis. So these are so we looked at the changes in the detections of the large-billed crow. This is the Japanese bush warbler, and this is the that same owl, the forest specialist Ryukyu scops owl. And so we modeled the changes in vocalization detections after the typhoons. So this, you're going to see a kind of center line down the middle here, And if the credible intervals here are not spanning 0, then we can say with some confidence that there was a change. If those distributions are to the left of the line and in gray, then it's showing a decline in the detections of that species after the typhoon. If they're to the right and in blue, it's an increase in the detections of that species after the typhoon. And so for the crow, we pretty much saw nothing across the sites. The crow seemed pretty unaffected actually by the typhoon overall, despite that very preliminary look on the previous data. And surprisingly to us also, the forest specialist owl was basically unaffected as well. But this Japanese bush warbler was pretty uniformly negatively affected by the typhoon. That is, we were detecting it less often, less frequently after the typhoon. And we can't say, of course, for sure whether that was mortality, behavioral change, or distributional change, but we certainly saw that there was fewer of them vocalizing in our acoustic data. And so finally, I'm just going to share the key result from our acoustic index analysis of this same project. So that was the species detections, this is the acoustic indices. And so across all 24 sites, and then also separately broken down by forested and developed sites, we looked at the variation among sites, just measured as the coefficient of variation at each point in time. So we did that, as I say, for everything and then separately for each one. And if we do this at multiple time points, we can generate these time series of spatial variability. So this tells us how different are the different field sites from each other in their acoustic fingerprint. And so what we were expecting to see, because of course these typhoons were really massive and were a real destructive force on the island, and we know anecdotally that the habitat was changed quite significantly in terms of destruction of vegetation, changes to canopy cover, and so on. So we expected there would be some standard differences among the sites before the typhoons, and that the typhoons would come through and basically homogenize the soundscapes a bit, because we expected almost a mass mortality event, lots of the soundscapes all simultaneously getting quieter at the same time. And so we expected this decline in how different they were from each other, because we expected them to converge on silence almost. And then we were expecting to see some kind of recovery afterwards. But what we saw was basically the complete opposite. So we saw that there was this divergence in how the different field sites were responding to typhoons in terms of their acoustic signature. So both when we looked at all the sites together and when broken down by forested and developed sites, in all cases, the sites got more different from each other after the typhoons, which is suggesting that the site level, even though we didn't see strong evidence of site level responses at the kind of regional scale across the island, we almost saw it. I don't want to call it emergence because that sounds dramatic, but we saw this kind of wider scale variation among the sites and how they were responding. And so we started talking about whether this could be an indicator that might be some capacity for maybe spatial insurance effects and this kind of thing, but it's a little hand wavy. But so to get a little bit less hand wavy, because these typhoons are getting more common, this was another big one that hit us in July last year, stranded some of you elsewhere. I think you guys were stuck in Tokyo, right? And so, this was another really nasty one. And so thanks to the really rapid organization of Masako Ogasawara and Juanita Chu, we've been taking canopy photos almost immediately after the typhoon and every few weeks since. And so what can we learn from this? Well, the first thing is that I clearly like circular photos of plants. But the second thing is that we can hopefully relate canopy recover then more directly to changes in the soundscape. And so the goal is to measure canopy recover and then regress that against soundscape change. There are, of course, other types of disturbance that I'm interested in and that you can use acoustics to think about. And this is a recent OIST COI Next project that we've just started with Sarah Keane and colleagues in Fukushima. And we're interested in, so after the 2011 Fukushima nuclear disaster, there was, the government established an exclusion zone, a pretty big exclusion zone, where people are no longer allowed to live. And so farmers basically abandoned their land and people left. And this is still mostly an exclusion zone today. And so what we're doing now is putting some acoustic monitors out, these much cheaper units, I should say. This is an audio mop, about 10 times cheaper than the one that you saw earlier. And we're putting a bunch of those out inside and outside the exclusion zone to look at the effects of land abandonment on biodiversity 10 years on from the disaster. And then finally, we also contributed to the Silent Cities project. So this was a very large scale and quite unique opportunity to do a natural experiment, quote unquote, based on the COVID-19 lockdowns. So during coronavirus, a lot of places around the world, people were sheltering inside and they weren't going outside. And so there was a big change to the presence of humans in the soundscape and in these otherwise normally urban environments, right? And so this project was with more than 250 authors, was a global effort to try and monitor changes in the soundscape as from the lockdowns and then back to normality afterwards to kind of infer what human noise pollution and anthropogenic activity can do to natural soundscapes. So hopefully from this second section, you've seen that we can use ecoacoustics as a tool to understand these disturbance impacts. And so finally, I'm just going to introduce the concept of response diversity, which is really what brings a lot of us here over the next few weeks. And this is what our TSVP thematic program is all about. So coming back to this, this idea about the insurance hypothesis, I'm really interested in moving not just from thinking about patterns, but towards these underlying biological mechanisms, asking, is there a general mechanism that drives this insurance effect of biodiversity across contexts? Or what is it specifically about biodiversity that is stabilizing? And that's a very big question. And so it takes a big team of people to try and get at any of these answers. So I should say the graphic is pretty out of date now, but the numbers are right. So Owen and I started the Response Diversity Network to try and kind of coordinate some research on response diversity specifically as a potential driver of stability, but also just stability mechanisms more generally. And we're really still just getting started. And so that's why opportunities like this thematic program are really, really essential to getting this moving. So I'm really excited for the next few weeks. So what is response diversity? Well, people have been thinking about it in ecology for about 20 or so years. So it's not exactly a new concept. And in this first paper, Elmquist and colleagues defined it as the diversity of responses to environmental change among species contributing to the same ecosystem function. So these three concepts come up in almost every definition. So we see it's something about the diversity of responses, something about environmental change or disturbance, and then a lot of them also have this like functional similarity or functionally equivalent species. But then in 2013, Akira Mori and colleagues dropped that need for those species to be functionally equivalent. It was just the diversity of responses in a community, basically. So what does that look like? Well, to vaguely and maybe a bit weakly tie this to Japan, I put up some sakura. So this is the Japanese cherry blossom, of course, and maybe we're interested because they provide a lot of really important cultural ecosystem services. Maybe we're interested in how their biomass is related to temperature or some other environmental change variable. Then under the insurance hypothesis, we know that if say these three different types of sakura, these different species or subspecies or whatever, are responding in exactly the same way to temperature, then if we see a pretty rapid or dramatic loss of a change in temperature, then we might see an equivalent loss of sakura across this whole community, which might have quite negative effects. Whereas in the middle, we've got an example where there's perhaps a low level of response diversity. That is, these species are all responding to temperature, but they're responding at different rates. So yes, they're responding in the same direction, but the rate of change is different with a change along the environmental x-axis, in this case, temperature. And finally, on the right, we've got a case of much higher response diversity, which is kind of akin to the portfolio effect or the insurance hypothesis that says that if you've got a decline in one species with, say, a change in temperature here, that decline is more likely to be offset by positive or neutral responses of other species if there is a greater diversity of those responses in the community. And so despite decades of interest in response diversity, based on those reviews, there really aren't that many empirical studies of response diversity today. We did a systematic review and we found about 50 or so people who were calling it response diversity and were agreeing with our definition of response diversity. And so we aimed to ask how people are measuring response diversity in practice. And we found that broadly, people were thinking about response diversity in two different ways. I'm going to focus on each in turn. The first is to think about these, what we were calling lower level traits. So these kind of typical functional traits of an organism. So you go out, you measure some aspect of an organism's trait. Maybe it could be its, it could be leaf area, it could be the number of eggs that a bird lays or something like this. And the idea is that that should then indirectly relate to some aspect of how that species should respond to environmental change. So for example, the number of eggs you lay is probably a good indicator of how well your population will bounce back after disturbance. And so these kind of lower level response trait-based approaches have been tied to stability of ecosystem functioning in several cases. So this is a nice example from Takahiro Sasaki and colleagues in 2019, where they looked at the response traits and the diversity of those traits in these Inner Mongolian plants. And they found that there was a link between the response diversity based on this measure and the temporal variability of these multiple ecosystem functions. But the strongest effect was not a direct link between response diversity and functioning. It was actually an indirect link via the effect of response diversity on the synchrony or asynchrony of different species populations among the different species. And then recently we found a pretty similar result in a completely different study system. So this is now, so the previous one was plants in Inner Mongolia. This is now birds in Romania. And this study was led by Hannah White. This was a lockdown project of ours. We did this analysis on response traits in birds, and we again found that they were related to the diversity, the stability of functional diversity or as a proxy for ecosystem function. But the stronger effect was again an effect via species asynchrony in a lot of cases. And so that really got me thinking about, well, exactly how response diversity might relate to these different aspects of stability. Remember, there's a lot of different ways of measuring stability. And so how should response diversity relate actually in practice to these different dimensions? And so we thought about this, and conceptually, there's a few people making cases for how response diversity should relate to different aspects of stability, such as temporal variability or resilience or so on. But there's really very little theoretical or empirical work evidencing those links still. And so I'm hoping that is something that we can work on in the next few weeks as part of this program is pushing towards, well, how should response diversity tie to stability actually, both mathematically and in empirical experiments and things. And so the other approach to measuring response diversity, which is what we call this higher level traits, these are typically kind of demographic or population level traits. So for example, you can see the abundance of a species change or its rate of population increase or something change along some environmental gradients. And then if you see that different species, the change of that value is different among different species, you can more directly measure the diversity of those changes. So we proposed that this approach that some people are doing, not as commonly as the previous one, should be a more direct measure of the diversity of different responses in the community. And so we generalized this to think of these as being performance-environment relationships. So where performance is any measure of individual fitness or contribution to ecosystem functioning, for example. So it could be abundance, it could be population growth rate, or some even like crop pollination and services or something. And the environment can actually, I mean, we think about this quite conceptually. It really doesn't matter what the environment is, but of course, individual people will have different interests. But it can be any biotic or abiotic environment. So you could be thinking about responses to temperature or land use change, or it could be something biotic like the predation pressure or competition instead. So what does that look like empirically? Well, we did a reanalysis of one of Owen's papers from 2009 with Dan Leary, where they had these 4 species of ciliates in Petri dishes, which, if you ask me, is just a really small bucket. And you had these four species responding in different ways. And what we first did to try and think about a way to get at the variety of these different responses is take the derivatives of these. The first derivative tells us the change in the slope at any given point along this x-axis. And so when we do that, it means that we get these temperature-dependent derivatives, which already was something different than what Owen and Dan's analysis was originally. And then from there, we can measure response diversity. And I'm actually not going to tell you how we measured response diversity, because I actually don't think it matters. And I also don't think it's perfect. I think there's a lot of work to be done around the specific measures of going from something like this to some summary of response diversity. I think we're far from there. But what I think was interesting to me, at least coming out of this analysis, is that because these derivatives varied along a temperature axis, the response diversity that we got out at the end also varied along a temperature axis. And so that meant that when we're thinking about the relationship between the environment and stability, say you've got some change in stability with temperature of some community, then it could be that overall change might be driven by some underlying biotic mechanism, such as a change in response diversity or some other driver with that temperature axis. And I don't know, it was just something I found interesting at the time. But we're now trying to extend this to, I think a natural step would be to extend this into multiple dimensions of environmental change, because of course, most ecosystems in the world are not just exposed to, say, temperature change. They also are exposed to these other global change drivers at the same time. And so this is something that Francesco Palazzo is leading in Owen's group. And we're asking how we can actually do this. And it is doable. It's a little bit complicated, but the answer is you need to build a multi-dimensional response surface, and then you need to know your position on the surface and the direction of environmental change on that surface. And then from there, you can actually quantify response diversity. And so hopefully you've seen that response diversity is a young concept in ecology. There's still a lot of work to do, and that's why I'm really so excited that we got this thematic session going. And I just can't believe so many awesome people said yes when I sent them this email. And so we've got some really, really cool talks coming up over the next couple of weeks. So please do come to these talks, interact with people, join us for lunch, take the time to meet with people, because there's a phenomenal set of people coming. So I'm just going to end there by saying that, hopefully you've seen that response diversity is a promising candidate mechanism for stabilizing communities and ecosystems, but we're still lacking both theoretical and empirical work on this. So I'm just going to stop there and thank, of course, everyone involved in these projects. Thanks. We're already running late, but I guess maybe 2 minutes for questions or something if anybody has. Don't feel obliged. Alberto?
Hello. Nice talk.
Thank you.
So I was trying to understand this insurance effect.
Mhm.
It seems to me that there were simulations where a sort of productivity parameters was defined.
Yes.
The claim was that productivity was higher and with lower variance
Yes, that's right.
as you increase the number of species. But how is this productivity defined in the simulation?
That is a great question, and I would need to read the paper again to tell you for sure. But yeah, Owen, you might know the answer.
It's totally abundance.
Total. Oh, it's just total. Oh yeah, it's maybe just total. Yeah, yeah, just like total abundance of everything in the. So you've got the different species, and it's just calculating the output of everything, basically. Yeah. Yeah. Sure.
You mentioned that Murray dropped along to the same function.
Yeah.
That's not something that I realized or thought about. Do you got any insights?
I've been thinking about this because we also dropped that when we wrote our methods paper, but I don't know, I don't think that these species necessarily need to be functionally equivalent for response diversity to matter for stability. If you're interested in the insurance effect and you're interested in the stability of functioning, then yes, I guess they should be functionally similar because they should be contributing to the same function, and therefore your response diversity should necessarily lead to a stability of the function. But I guess if you just care about community biomass, I don't know, I don't think they need to be functionally equivalent because you're just saying like, what are the species in the community? Are their different responses able to stabilize some output? I don't think it matters.
Doesn't that? Sorry.
Yeah, go on.
I mean, you just defined your function as biomass.
Well, okay, yeah, that's true. Yeah, that's a valid point.
So is there another way that you could avoid that sort of circularity?
Yeah. Yeah, that's a great, yeah, I guess. But even if you say just like total abundance at the community level, that's still an aggregate output, right? Maybe they. I think that's one to think on. Yeah, sounds good. Pass?
I just kind of a random thought, but like I feel like, especially when you do about response diversity and COVID and things like that, there seems to be a lot of analogies you could draw to like economics and like small companies and how they responded.
Mm-hmm.
We had a talk a while ago about Japan's like diversity of like little companies compared to America's
Mm-hmm.
like big giants, and I wonder whether the fields can learn something from each other.
That is a really spot on point, actually, because the insurance effect, also the portfolio effect, which is people use a lot in ecology, has been adapted from finance, from economics. And a lot of these, Michelle Leroux and Laura, who's coming, and a bunch of people wrote this really nice, maybe some people are here, sorry if you're on that paper, wrote this really nice review that basically actually made a direct link saying, okay, well, here are all the concepts about diversity, stability, and ecology, and here's how they relate to economics. And it's a really cool paper, actually. Yeah. If you're interested in that kind of stuff, that's out there. But yeah, a lot of these ecologists like to borrow ideas from other fields. We absolutely love taking mathematicians and physicists, particularly, and turning them into ecologists. And yeah. But anyway, yeah. Thanks. I guess maybe we should move to Owen's talk more. We're of time. Yeah, shall I? Okay, I guess I should take this one. Thank you.